guys. So today we'll be studying about a DNA motif, that is helix turn helix. So like previously we studied about the transcriptional activators, and these transcriptional activators, they're divided into two, that is the DNA binding domain and the transcriptional activating domain. The DNA binding domain is also called as motifs. You have the zinc containing modules, Homeobox domain, and bzip and bhlh motif. So in that, the zinc containing modules will be taught to you guys by Rakshita. It's a minor seminar on Friday. So, and today we'll be focusing on the Homeobox domain, that is the helix turn helix. So coming to this Homeobox domain, so this Homeobox domains, it was found in a large family of activators, and the name comes from a gene region called as the Homeobox, and it was discovered in regulatory regions of the fruit flies in Drosophila, which is also called as the homeotic gene. So how do you find a function of a gene? So you find a function of a gene by mutating them. So when you mutate, and these mutations of that homeotic genes found that they cause transformations of the body part of the fly. For example, here you can see in the picture. So here you can see the anapedia antinapedia mutation. So when antinapedia gene was mutated, it was shown that the legs will start growing in the regions where antennas are supposed to grow. And the Homeobox domain, proteins, they form the member of the helix turn helix family of the DNA binding protein, and each Homeo domain has 3 alpha helixes. The second and the third, they form the helix turn helix motif, and the third will form the recognition with the third, which forms the recognition helix. And it also has an N terminal which will go and bind to the minor groove of the DNA. So here in this picture you can see the Drosophila. Here you can see the Drosophila homeotic gene engraved, which is bound to its target DNA. And the recognition site, this is the recognition site which is given in red color. And then you also have the N terminal. So this N terminal is attached to the minor groove of the DNA. And here you can see these are the 2 alpha helixes which forms the helix turn helix. So coming to summarize the homeodomains, so these are found in the eukaryotic activators. They contain a DNA binding motif which functions in the same way as helix turn helix motif, and the recognition site will bind to the major groove and make specific contact there, whereas the N terminal, it will go and bind to the minor groove. Now going to the helix turn helix motif, it is one of the most simplest and the common DNA binding motif, and it is the first DNA binding motif to be recognized was the helix turn helix, and it was first identified in case of the bacteria, and now it is also found in both prokaryotes as well as the eukaryotes. So this helix turn helix, it is made up of 2 alpha helix. So here the diagram it just shows how the gene regulatory protein, like in general the binding of the gene regulatory protein to the major groove, like we have studied in the previous video, that most of this DNA motifs, the main target region is the major groove. So this diagram just shows how it will go and bind to the major groove, and these are the different bases which are present. So now coming to this helix turn helix. So this helix turn helix, it is made up of 2 alpha helixes. So I know you guys have not studied about the alpha helix or the beta sheet in detail, because we'll be studying about that in a chapter later in the semester itself. So for now, what you have to know is that alpha helix, they have this they look like this. These are the alpha helix, and then you have beta sheets. So these they are the secondary structures of the protein. So in helix turn helix, what you find is that it is made up of two alpha helices, which is connected. So these two alpha helices, they're connected by a short chain of amino acids, which will constitute the turn. So you have these two regions, and these two alpha helices, they are attached at a fixed angle, and you have this C. This is called as the C terminal. The C terminal, it forms the recognition site, and this it will go and bind to the major groove. Whereas you have the N terminal, it will go and bind to the minor groove. And then coming to the different types of helix turn helix. So you have the dihelical. So it is the same example as the engrail, which we already discussed in the previous diagram. And then you have trihelical. Example is transcriptional activator MYP. So here you can see the trihelical. Here you can see the trihelical, where there are three alpha helixes. So it is called as the trihelical. And then you have the tetrahelical. Example for tetrahelical is Lexa type DNA binding HTG domain in the bacterial transcriptional factor TER receptor. And in tetrahelical, the difference is that they have an additional C-terminal helix compared to the trihelical motif, and it is found in these regions. And then you have the winged helix turn helix. So in winged helix turn helix, you can see in the winged helix turn helix, it is made up of you can see here H1, H2 and H3. So the winged helix turn helix is made up of three helical bundle, and the third or the fourth strand, it will form the beta sheet. So this purple color refers to the beta sheet, and then you have three green helical region, and these are found in the transcriptional factor ETS. So that is all about the different types of motifs, the different types of the helix turn helix motif. And coming to the location, so where are these present? So they're present in all the eukaryotic proteins, and also the bacterial regulatory proteins, which are related to the eukaryotic proteins. And the main function is to bind to the DNA and regulate the gene expression by binding to the major groove through a series of hydrogen as well as van der Waals interactions. I hope you guys understood. And if you have any doubts, feel free to ask me. And in the next video, we'll be focusing on the B-zip and the BHLH motif. In that, we have two DNA binding motifs, that is leucine zipper as well as helix loop helix. Thank you. Voice hug.